Hello everyone and welcome back to our channel MooTechie. Today, we're diving into one of the most fundamental and beautiful molecules in all of biochemistry, DNA. We'll be exploring its structure, its functions, and its fascinating properties. Whether you're a medical student or just have a curious mind, this journey to the center of life's code is going to be exciting. Let's get started. First, let's define what DNA is. DNA is a polymer of deoxyribonucleotides. It's found in chromosomes, mitochondria, and chloroplasts. And most importantly, it carries the genetic information. To understand DNA, we must first understand its building blocks. So what are the components of a nucleotide? They are a base, a sugar, and a phosphate group. Let's visualize this. A nucleotide is formed when a phosphate group attaches to a nucleoside. And a nucleoside itself is just a base attached to a sugar. The key difference between DNA and RNA is in this sugar. If X is a hydrogen atom, it's DNA. If X is a hydroxyl group, or OH, it's RNA. Now the bases themselves come in two fundamental types, which are like the architectural blueprints for our genetic alphabet. We have pyrimidine, wood, which is a single six-membered ring. Its structure is shown with nitrogen atoms at positions 1 and 3. And we have purines, Re, which is a larger double-ring structure with a six-membered ring fused to a five-membered ring, and nitrogen atoms at positions 1, 3, 7, and 9. Let's put names to these structures. The pyrimidines include cytosine, thymine, and uracil. The purines are adenine and guanine. Now, how do we name the full components? This is where nomenclature comes in. We have the base, which forms a nucleoside when attached to a sugar, which in turn forms a nucleotide when a phosphate is added. Many nucleotides together form a nucleic acid. For the purines, adenine becomes adenosine in RNA and deoxyadenosine in DNA. Their nucleotides are adenylate and deoxyadenylate. Guanine becomes guanosine in RNA and deoxyguanosine in DNA. Their nucleotides are guanylate and deoxyguanylate. For the pyrimidines, cytosine becomes cytidine in RNA and deoxycytidine in DNA. Their nucleotides are cytidylate and deoxycytidylate. Thymine is found almost exclusively in DNA, so it's thymidine or deoxythymidine, and its nucleotide is thymidylate. And uracil is found in RNA, forming uridine and uridylate. This isn't just academic. Understanding these structures has saved lives. Nucleoside and base analogues can be used as anti-cancer and antivirus drugs. For example, 5-fluorouracil and 6-mercaptopurine are powerful anti-cancer agents. And drugs like azidothymidine and dideoxynosine are crucial antiretroviral agents used to treat viral infections like HIV. Now, let's build these nucleotides into the magnificent structure of DNA. The primary structure of DNA is simply its sequence, the order of the bases. It has directionality. We have a 5' end and a 3' end. The nucleotides are linked together by phosphodiester linkages between the 5' phosphate of one nucleotide and the 3' hydroxyl group of the next. We can depict this as a chain: 5' end, then a phosphate, then A, then phosphate, C phosphate, G phosphate, T phosphate A, and finally an OH at the 3' end. Traditionally, a DNA sequence is drawn from the 5' to the 3' end. So, a shorthand notation for this sequence is simply ACGTA. But DNA isn't a flat string. It has a stunning three-dimensional shape. The secondary structure of DNA is the famous double helix. So what defines this double helix? It consists of two anti-parallel polynucleotide chains wound around the same axis. One chain runs 5' to 3, while the other runs 3 to 5. The sugar-phosphate chains wrap around the periphery, forming the backbone. And the bases A, T, C, and G occupy the core, forming complementary A with T and G with C Watson-Crick base pairs. The DNA double helix is held together mainly by hydrogen bonds. A hydrogen bond is a chemical bond in which a hydrogen atom of one molecule is attracted to an electronegative atom, like nitrogen or oxygen, usually of another molecule. This bonding is specific. There are two hydrogen bonds between an A and a T base pair. And there are three hydrogen bonds between a C and a G base pair. This is why GC pairs are stronger and more stable than AT pairs. But there's another often overlooked force holding the helix together: base stacking. The bases in DNA are flat and planar, and they have a tendency to stack on top of each other like a pile of coins. The major stacking forces are hydrophobic interactions and van der Waals forces. This stacking is crucial for the stability of the double helix. Now, DNA is not a one-size-fits-all structure. It can take on different forms. Here is a comparison of the main structural forms: aDNA, bDNA, and zDNA. Looking at the table, helix-handedness, both A and B DNA are right-handed, while zDNA is left-handed. Base pairs per turn, aDNA has 11, bDNA has 10.4, and zDNA has 12. Rise per base pair, aDNA is 0.23 nanometers, bDNA is 0.34 nanometers, and zDNA is 0.38 nanometers. Pitch, aDNA is 2.46 nanometers, bDNA is 3.40 nanometers, and zDNA is 4.56 nanometers. Diameter, aDNA is 2.55 nanometers, bDNA is 2.37 nanometers, and the slimmer, zDNA is 1.84 nanometers. Glycosidic bond A and B DNA have an anti-conformation, while zDNA alternates between anti and syn. Major groove, it's present in A and B DNA, but absent in zDNA. The most common form in our cells, the normally hydrated DNA, is the B form. Let's look at a diagram of bDNA. It's a right-handed helix. The base pairs are almost perpendicular to the helix axis and are 3.4 angstroms apart. One full turn of the helix is 36 angstroms and contains about 10.4 base pairs. You can see the major groove A, which is about 22 angstroms across, and the minor groove A, which is about 12 angstroms across. These grooves are vital for proteins that read the DNA sequence. In the complex world of eukaryotic cells, DNA isn't just floating around, it's highly organized. It is folded into a structure called chromatin. This brings us to the DNA tertiary structure. The DNA double helical structure coils around proteins called histones. DNA bound to histones form structures called nucleosomes, which look like beads on a string and are known as 10 nanometer fibers. Each nucleosome core contains about 146 nucleotides of DNA. So what is a nucleosome? It's one of those repeating globular subunits of chromatin. It consists of a complex of DNA and a histone core, with linker DNA connecting one nucleosome to the next. The core itself is made up of a protein octamer. Imagine a core of 8 histone proteins, 2 copies each of H2A, H2B, H3 and H4. The DNA is wrapped around this protein core like thread around a spool. Now the compaction doesn't stop there. To fit an enormous amount of DNA into a tiny nucleus, it undergoes incredible levels of packaging. The beads on a string form coils to form a 30 nanometer fiber. This fiber then forms loops, each containing about 75,000 base pairs. About 6 loops form a rosette. 30 rosettes form one coil, and 10 coils form one chromatid. A chromosome consists of 2 such sister chromatids. This is an amazing feat of biological engineering. Sometimes DNA can form a supercoil, which is essentially a coil over a coil. The diagram shows a relaxed circular coil, and then the same coil twisted upon itself into a supercoiled state. This supercoiling helps in packing and also plays a role in processes like transcription and replication. Now, let's talk about the behavior of DNA in the lab and in the cell, DNA melting and annealing. DNA can undergo reversible denaturation and annealing. We start with double helical DNA. When we apply heat or certain chemicals, it denatures or melts. The hydrogen bonds break and the strands separate into partially denatured DNA and then fully separated strands in random coils. But if we then slowly cool the DNA, the complementary strands can find each other again and reassociate or anneal by base pairing, reforming the double helix. A key concept here is the melting point, or T_M of DNA. This is the temperature at the midpoint of the transition from double-stranded to single-stranded. On a graph, you would see a sigmoidal curve with that _M right at the 50% denaturation point. So what does the T_M of DNA depend on? It depends heavily on the content of G and C base pairs. Since GC pairs have 3 hydrogen bonds, DNA with more GC content has a higher melting point. A graph shows this clear positive relationship. As the percentage of G plus C increases, that _M also increases. It also depends on the size of the DNA molecule, the pH, and the ionic strength of the solution. Finally, let's summarize the functions of DNA and its structure. DNA consists of 4 bases, A, G, C and T, that are held in a linear array by phosphodiester bonds through the 3' and 5' positions of adjacent deoxyribose sugars. DNA is organized into 2 strands by the pairing of bases, A to T and G to C on complementary strands. These strands form a double helix around a central axis. In humans, the roughly 3 billion base pairs of DNA are organized into the haploid complement of 23 chromosomes. And most importantly, DNA provides a template for its own replication, ensuring the genotype is maintained. It also serves as the template for the transcription of our roughly 30,000 genes into a variety of RNA molecules, which ultimately direct the synthesis of proteins that run the cell. And that, everyone, is the story of DNA, from its simple chemical components to its elegant double helix and its crucial role as the blueprint of life. Thank you for learning with me today. See you in the next class.